So we're back with Enzymes Part 2 for my biology students. I must apologize that it's taken so much and it's taken a while for me to make this one, but I had a lot of work going on. But of course, I found the time to make this because it'll be a great advantage to you to not only know the effect of temperature on enzyme activity, but also the effect of the pH on enzyme activity, as it is, of course, required in the syllabus of grade 10, 11 ordinary level biology. Anyways, so the effect of pH is very much different from that of temperature. So on temperature, we notice that when the temperature is lower, well, the enzyme works at a very slower pace, at a lower energy level, compared to when it is very high. When the temperature is very high, now the enzyme has denatured and the bonds which held it in a solid shape have been broken, resulting in the active site being changed and then there being no reaction because now the substrate can't fit in to the active site. But if you can't understand anything I'm saying here based on temperature, I suggest please go watch the video on enzymes part 1, enzyme specificity, and the effect of temperature on enzyme activity. Almost lost my voice there, but water is good. What are we without water? Anyways, moving on. So, here we see that the effect of pH on enzyme activity is such that an enzyme will only work at a specific pH only. So it cannot work neither low nor above, but at those specific narrow limits. Let's say if it's enzyme amylase, which we see right here, um, then it will only work at pH 7 to, let's say, 7.5. So there are very narrow limits when it comes to pH, okay, as compared to temperature where we have, when it's lower, let's say it's at 2, it'll go, it'll work for a slower pace, but then when it gets too high, it denatures. Anyways, moving on. So enzymes are specific to the pH. That means they'll only work at a specific pH. Enzymes will only catalyze reactions at a certain specific pH. It can neither be low nor higher. And as you can see, pepsin only works at pH 2. Amylase only works at pH 7. And lipase only works at pH 13. And that means we have narrow limits. That means pepsin could work from pH 2 to pH 2.5 perhaps, and amylase can work from pH 7 to pH 7.5. The moment the pH hits 8, no reaction from amylase. Alright? One thing you need to know, there are very narrow limits when it comes to enzyme reactions in regards to the effect of the pH on the enzyme activity. Lipase can work at pH 13 only, and perhaps narrow limits whereby we have pH 13.5, perhaps, yes. But the moment the pH hits 14, there's no reaction. Okay? And if the pH hits 12.9, there's no reaction. It's very specific to the pH, right? Because the pH determines the conditions in which this enzyme will work. For instance, pepsin, now pH 2 is acidic, right? pH 2 is acidic. I'll have it written down here so you guys can see. But pH 2 is acidic, pH 7 is neutral, and pH 13 is alkaline. So that means that pepsin works best in acidic conditions. That's why any higher or lower pH level would not work for pepsin. The same goes for amylase. Amylase only works in neutral conditions. That means any pH lower or above would be what? Acidic or alkaline, which would, not correlate with the requirement for amylase to be able to catalyze reactions. Same goes for lipase. Lipase only works at pH 13, which is an alkaline condition, an alkaline environment. Therefore, any pH level that is lower than pH 13 could be acidic. Okay, and that is why it will not work. So, pepsin digests proteins to polypeptides. Amylase digests starch to maltose, and lipase digests fats to fatty acids plus glycerol. Now, you might not know what these mean, so I suggest you go watch the video on biological molecules, which gives a brief overview. Excuse me for that. A brief overview on fats, proteins, and carbohydrates. But just to help you clarify a little bit, proteins are biological molecules which help our bodies to function, to make new cells for cell growth and cell repair, but also it's the main component of the antibodies and, of course, the hemoglobin, which is the protein that is contained in the red blood cell, used to bond with oxygen to transport oxygen around the body. Again, you might want to just go watch another video on that. All right, perhaps I'll make one on transport of blood around the human body. Anyways, moving on. So for amylase, starch is a carbohydrate, but a very complex insoluble carbohydrate, which is a sugar. All right, a complex sugar, to maltose, which is a simple sugar. So amylase will break down this complex sugar to a simple sugar. Same applies for same applies for pepsin, which will digest protein, which is a complex molecule, to polypeptides, which are a very simple chain of amino acids. Lipase digests fats to fatty acids plus glycerol, which are the building blocks for all fats and lipids. Alright, so we are done with this section. I might as well just end it here before we have a limit that cuts short what I might have to say next.